Hello friends, welcome back to my YouTube channel Journey with VIJAYKUMAR SHRIVASTAV. Today I am going to present the topic photoperiodism. Before presentation, little introduction about myself. I am VIJAYKUMAR SHRIVASTAV. I have done M.Sc Agriculture with specialization in Agronomy from Pant University of Agriculture and Technology, Pantnagar in 1996 and presently working in seed industry and associated with one of the M.Sc. So let's proceed to the presentation. What is photoperiodism? Photoperiodism is the phenomenon of physiological changes that occur in plants in response to relative length of day and night, like photoperiod. The response of the plants to the photoperiod expressed in the form of flowering is also called as photoperiodism. The phenomenon of photoperiodism was first discovered by Garner and Allard in 1920. Depending upon the duration of photoperiod, the plants are classified into three categories. First one is short day plants, second is long day plants, and third is day neutral plants. First one is short day plant. These plants require a relatively short day light period, usually 8 to 10 hours per day, and a continuous dark period of about 14 to 16 hours for subsequent flowering. These plants are known as long night plants, like rice, coffee, soybean, tobacco, and chrysanthemum. In this diagram, we have shown the flowering behavior of short day plants. In first case, when day period is less than 12 hours and dark period is more than 12 hours, there will be flowering. In second case, when day period is less than 12 hours and there is gap between dark period, means in between there is a gap of light, then flowering will be affected and it will result as no flowering. In third case, when the day period is less than 12 hours and there is prolonged dark period for a long time, again in between if there is light period and again dark period, then in such case also there will be flowering. And fourth case, when the day period is less than 12 hours and beginning of the dark phase there is interruption of light and again for longer period there is dark period, then there will be also flowering. In short day plants, the dark period is critical and must be continuous. If this dark period is interrupted with a brief exposure of red light having 660 to 665 nm wavelength, the short day plant will not flower. Maximum inhibition of flowering with red light occurs at about the middle of critical dark period. However, the inhibitory effect of red light can be overcome by a subsequent exposure with far red light having wavelength of 730 to 735 mm. Interruptions of the light period with red light does not have inhibitory effect on flowering in short day plants. Prolongation of the continuous dark period initiates early flowering. In this diagram, in short day plants, we have shown the interruption of red lights and far red lights impact on flowering behavior. Second plant is long day plants. These plants require longer day light period, usually 14 to 16 hours, in a 24-hour cycle for subsequent flowering. These plants are also called as short night plants, like wheat, radish, cabbage, sugar beet, and spinach crops. In long day plants, light period is critical. A brief exposure of red light in the dark period or the prolongation of light period stimulates flowering in long day plants. In this diagram, we have shown the flowering behavior of long day plants. If day period is more than 12 hours and dark period is less than 12 hours, there will be flowering. Second stage, when the day period is less than 12 hours and the dark period is more than 12 hours, then there will be significant impact on flowering and there will be no flowering. In third case, when the day period is less than 12 hours and during dark period there is interference of light for a shorter period, in such case also there will be flowering. Third type of plants are day neutral plants. These plants flower in all photoperiod ranging from 5 hours to 24 hours continuous exposure to light, like tomato, cotton, sunflower, cucumber, peach, and certain varieties of tobacco. During recent years, intermediate categories of plants such as long short day plants and short long day plants have been also recognized. About long short day plants, these plants are short day plants but must be exposed to long day during early period of growth for subsequent flowering, like Bryophyllum crop. Second is short long day plants. These are long day plants but must be exposed to short day during early periods of growth for subsequent flowering, like certain varieties of wheat and rye. Here we will go through the table which is showing the photoperiodic response of some common plants. First, short day plants. There are some important crops like chrysanthemum, cocklever, cosmos, dahlias, goosefoot, hemp, morning glory, poinsettia, rice, soybeans, tobacco and violets. And out of long day plants, some important crops like barley, cabbage, carrot, henbane, larkspur, lettuce, onion, petunia, poppy, radish, spinach and wheat crops. And among day neutral plants, some important crops are balsam, beans, chilies, cotton, cucumber, dandelion, Jerusalem artichoke, maize, potato, tobacco and tomato crops. Here we have shown the chart in which we will show the vegetable crops responses to photoperiodism. First one, day neutral vegetable crops like tomato, pepper, eggplant, cucurbitaceae, vegetable crops, cowpea, okra, French bean and amaranth. In long day plants, there are some important vegetable crops like potato, onion, lettuce, cabbage, cauliflower, radish, spinach, beet, turnip and carrots. And among short day plants, the important vegetable crops in this category is sweet potato, Indian spinach, hyanthus beans, cluster bean, winged bean, etc. Now we will study about differences between short day and long day plants. First, in case of short day plants, plants flower when photoperiod is less than the critical day length, while in case of long day plant, plants flower when photoperiod is more than the critical day length. Second, in case of short day plants, interruption during light period with darkness does not inhibit flowering, while in case of long day plant, interruptions during light period with darkness inhibit flowering. Third, in case of short day plant, flowering is inhibited if the long dark period is interrupted by a flash of light, while in case of long day plant, flowering occurs if the long dark period is interrupted by a flash of light. Fourth, long continuous and uninterrupted dark period is critical for flowering, while in long day plants, dark period is not critical for flowering. Fifth, in short day plants, flowering does not occur under alternating cycles of short day and short light period, while in long day plants, flowering occurs under alternating cycles of short day followed by still shorter dark periods. Phytochrome. It is observed that a brief exposure with red light during critical dark period inhibits flowering in a short day plant, and this inhibitory effect can be reversed by a subsequent exposure with far red light. Similarly, prolongation of the critical light period or the interruption of the dark period stimulates flowering in long day plants. This inhibition of flowering in short day plant and stimulation of flowering in long day plants involves the operation of a proteinaceous pigment that is called phytochrome. It is present in the plasma membrane of cells and it has two components, chromophore and protein. Phytochrome is present in roots, coleoptiles, stems, hypocotyles, cotyledons, petioles, leaf blades, vegetative buds, flower tissues, seeds and developing fruits of higher plants. The pigment phytochrome exists in two different forms. First one is red light absorbing form, which is designated as PR, and second is far red light absorption form, which is designated as PFR. These two forms of pigments are photochemically interconvertible. When PR form of the pigment absorbs red light, which is in the range of 660 to 665 nm, it is converted into PFR form. When PFR form of the pigment absorbs far red light having the wavelength of 730 to 735 nm, it is converted into PR form. The PFR form of pigment gradually changes into PR form in dark. About PR and PFR, it is considered that during daytime the PFR form of the pigment is accumulated in the plants which are inhibitory to flowering in short day plants but is stimulatory in long day plants. During critical dark period in short day plants, this form gradually changes into Pfr form, resulting in flowering. A brief exposure with red light will convert this form again into Pfr form, thus inhibiting flowering. Reversal of the inhibitory effect of red light during critical dark period is SDP by subsequent far red light exposure is because the Pfr form after absorbing far red light will again be converted back into Pr form. Prolongation of critical light period or the interruption of the dark period by red light in long day plants will result in further accumulation of the Pfr form of the pigment, thus stimulating flowering in long day plants. In this chart, we will study about Pr form and Pfr form of pigments. First one, Pr form is blue-green in color while Pfr form is light green in color. Second, in case of Pr form, it is an inactive form of Phytochrome and it does not show Phytochrome mediated responses. While Pfr form, it is an active form of Phytochrome and hence shows Phytochrome mediated responses. Third, Pr form has maximum absorption in red region, about 680 nm wavelength, while Pfr form has maximum absorption in far red region, about 730 nm. Fourth, Pr form can be converted into Pfr form in red region when the wavelength is 660 to 665 nm, while Pfr form can be converted into Pr form in far red region with wavelength 730 to 735 nm. Fifth, Pr form is found diffused throughout the cytosol, while Pfr form is found in discrete areas of cystosol. Sixth, Pr form contains many double bonds in pyrrole rings, while Pfr form contains rearranged double bonds in all pyrrole rings. Now we will study about significance of photoperiodism. Photoperiodism is an example for physiological preconditioning. The stimulus is given at one time and the response is observed after months. Exposure to longer photoperiods hastens flowering, like in wheat, the earring is hastens. During long light exposure, Pr form is converted into Pfr form and flowering is initiated. In dark period is greater, Pfr is converted into Pfr form that inhibits flowering. The important Phytochrome mediated photoresponses in plants include photoperiodism, seed germination, sex expression, bud dormancy, rhizome formation, leaf abscission, epinasty, flower induction, protein synthesis, pigment synthesis, oxygen catabolism, respiration and stomatal differentiation. So this was all about photoperiodism in plants. Now my presentation ends here. Thank you very much for your kind attention. See you again shortly. Here I have given my YouTube channel details. Journey with VIJAYKUMAR SHRIVASTAV. Having request, please visit the channel, subscribe it and provide your valuable feedbacks for further improvements. Thanks.